Welcome back to the Center. Today we're going to be talking about brain-to-computer interfaces with Dr. Leigh Hochberg and Dr. Dan Rubin. Dr. Hochberg is a vascular neurologist and neurointensivist at Massachusetts General Hospital. He's the IDE sponsor investigator and director of the BrainGate clinical trials. He's the senior lecturer on neurology at Harvard Medical School. He's an associate director at the VA RR&D Center for Neurorestoration and Neurotechnology. He's the L. Herbert Ballou University Professor of Engineering and Professor of Brain Science at Brown University. Dr. Daniel Rubin is a neurointensivist at Massachusetts General Hospital. He's an assistant professor of neurology at Harvard Medical School and a principal investigator for the BrainGate clinical trials. Dr. Hochberg and Dr. Rubin, is it okay if I call you Leigh and Dan for this episode?
Please do. It's good to see you, Josephine.
It's great to see you too. I have a lot of questions about brain-to-computer interfaces. So can we start by talking about BrainGate? Leigh, what is BrainGate?
BrainGate, that phrase, when we use it, means a few different things. So one is the ongoing BrainGate clinical trials, which started back in 2004 and continue to this day. BrainGate sometimes is also how we refer to the system itself, more formally the BrainGate Neural Interface System, which is an investigational medical device. That's the actual device or system that's been tested in the BrainGate trials. And then we think about our BrainGate Consortium, which is the really incredible group of people and their institutions that work so closely together to conduct this clinical trial with this particular investigational medical device. And so the BrainGate Consortium includes folks here where we're sitting at the moment at Mass General Hospital, also at Brown University, the Providence VA Medical Center, Stanford, Emory, and the University of California, Davis. So it's that group that makes up the consortium. And depending on the inflection in my voice, BrainGate means one of those three things.
Great, thank you. Dan, so Leigh mentioned the investigational medical device. From my understanding and from a very high level, the device can be separated into a sensor, a decoder, and an effector. Could you kind of summarize what these terms mean and really what is the sensor and the device as a whole?
Absolutely. Great question. So any brain-computer interface needs to have those three components in order to be a complete system. Brain-computer interfaces work in general by measuring continuously some neural signals that are associated with the intended or attempted movement or action of the person using it. So you measure those signals. You need a sensor to do that. Then you need a decoder, and this is really, to some extent, the heart of the engineering problem, is finding a one-to-one relationship between those neural signals that you measure, which are just electrical signals that you're recording in real time, and what the person is attempting or intending to do. So you need to find really a mathematical relationship between those signals and the user's attempted action. And then the last bit is the effector or the actuator. And that's sort of the most exciting part of BCI research. That's what you actually do with that signal. So the user's attempting or intending to move their arm or to speak, you decode those signals. You now know what they're trying to do, and the effector is the way that you help them do that. And so each of those three parts, when put together in the right way, can give you a brain-computer interface.
You know, when you said that one of them is the most exciting part, we automatically get 2/3 of the people mad at us.
Sorry, that's true. No, there's a lot of excitement around the sensors, and truthfully, being honest, I actually find the decoding part the most exciting myself. But for a lot of people, it's the actuators. That's true.
Okay, well, let's start by talking a little bit more deeply about the sensors themselves. So we're aware that neurotechnologies come in multiple flavors. There's invasive, there's non-invasive neurotechnologies. Why does the BrainGate array have to be intracortically implanted, Leigh?
So let me start because I happen to have one here with me. This is what's known as the Utah array. It's manufactured by companies, Salt Lake City, BlackRock Neurotech, but it was developed in the early 1990s by Dick Norman at the University of Utah, and then subsequently through a history of bionic technologies and then cyberkinetics and then BlackRock became available at various stages for use in investigational medical device research. So this is the device. I realize it's nearly too small to see from the distance I am at the camera at the moment, but this is the part where the action happens. It is this 4x4 mm array that is tapped in to the top of the brain that allows for the recording of dozens, if not 100 or more, single neurons. That is that all-important cell in the brain that has so much information encoded in it. We're able to record, we and others are able to record dozens, if not more, of individual neurons from each of these arrays that are tapped into the brain. So the reason that we use that particular device and have been using that particular device is largely because of the information that's encoded there. Neurons are the source of information inside the brain. There's how many neurons are in the brain according to your textbook?
88 billion.
88 billion. All right, good. So that's well within the range of the somewhere between 80 billion and 100 billion that we hear about all the time. And and so that's the number of neurons that are in the human brain. If we're actually able to record even just a handful, meaning a few dozen or maybe even a couple hundred of those, we can extract an extraordinary amount of information because it's when one neuron talks to another neuron, or a neuron from the spinal cord talks to muscle. That information content is what enables cognition or mood or memory or movement and so many of the other activities in the nervous system. So if we can extract that information, then hopefully we can use that information to restore communication and restore mobility for people with severe speech or motor impairments.
So Leigh, you mentioned people with motor impairments using these devices. How does the brain activity of an able-bodied person differ from someone with motor impairments?
So when I think about this field, I think about the clinical trials that we started in 2004, but I really think about the nearly 5 decades of fundamental, publicly funded basic research happening in university laboratories that led to that moment. That basic research really was focused on not only developing technologies that would allow for the recording of individual neurons in the brain, but also trying to understand how do different parts of the brain, how do individual neurons in the brain relate to the control of voluntary movement. So it became well known from years of research that if one was able to record chronically from, for example, the precentral gyrus or the motor cortex, that one could look at individual neurons, and sometimes if one was listening to one neuron and, for example, an animal in a laboratory was moving its forelimb or its arm to the right, that neuron might be firing a lot. And that same animal is moving its hand to the left, that neuron might not be firing so much. And up or down, the neuron was kind of firing at some in-between firing rate. So if all we knew about the hundreds of millions or billions of neurons in that particular brain, if all we knew at the moment was the firing rate of one neuron, we would have some idea, far from perfect, but some idea whether, again, in that 5 decades of research, that animal might have been attempting or actually moving its hand to the right or to the left. So that concept is known as the directional tuning of a neuron. There's a particular simple mathematical equation that can be fit to relate the firing rate of that particular neuron to the likelihood of its moving its hand in a particular direction. And that core research, where one began to understand the relationship between a neurologically intact animal in a university research lab and its brain activity, it was from that we were able to move, back to the question that you were asking, to be able to do clinical research towards helping people with paralysis. And what's been really both remarkable and in many ways exactly what was expected is that the types of neural activity that I've seen over the years, I'm sure that Dan has seen over the years, that we've seen recorded from, for example, the motor cortex of somebody who has cervical spinal cord injury, that is somebody who's unable to move their arms or legs. That neural activity looks familiar in many ways when compared to the decades of fundamental neuroscience research that had been done beforehand. So you asked how does the neural activity of somebody with paralysis relate to the neural activity of somebody who may be able-bodied? And while we don't have that exact comparison, what I believe is that those activities are very similar. And because they're so similar, not only were we able to take what is learned in the basic university laboratory and translate it to human clinical research, but also the research that we're doing, and I suspect Dan will be able to speak to this more in a moment, when we're learning and when we're recording these neural activities in the brain of somebody in our clinical trial, we're learning not only about how this part of the brain works in somebody who may have a disorder that leads to paralysis, but also we're learning about the fundamental human brain activity that exists in people who are able-bodied and people in any number of other conditions, not just the specific injury illness that may have led that person to join us in the trial.
And Dan, how do you decide where to place arrays in the brain?
That's a great question. And I brought a visual aid. So when someone wants to join us in the trial, and they decide to do so, before we bring them here to the hospital to place the arrays, we have to do some imaging. So the key to really all of neuroscience is similar to that of real estate: location, location, location. And we've known now for well over 100 years that different parts of the brain have different functions. And early work in the 1930s by neurosurgeons, where they would stimulate individual parts of the brain. This is Penfield's famous work. Would stimulate different parts of the brain as he was doing epilepsy surgery, and he would see that people would have a twitch of a finger, or if it was an awake surgery, would even say, "Hey, I feel something funny in my left hand." And this led to the earliest maps. They were called a homunculus, which literally means a small person, but you could literally see someone's body, if you will, laid out over different parts of the brain. Parts of the cortex controlling sensation to the hand were anatomically distinct from parts of the brain controlling movement of, say, the other arm. Long-winded way of saying that different parts of the cerebral cortex have different functions. Nowadays, we don't zap people's brains with little electrodes to determine what part of the brain controls what. We do imaging. So we do a lot of fMRI imaging, so functional MRI. So they come into the hospital. We bring them to the MRI suite, and we do a scan. From that scan, we're able to use some sophisticated mapping techniques where we look at what's called the functional connectivity. So how the blood flow to different parts of the brain changes over time in relationship to the blood flow to other parts of the brain, either while the person is resting or while they're asked to do a specific task, like try to open and close your hand, try to speak, try to fill in the blank of the sentence. And by looking where the blood flow changes as they do these different tasks, we can figure out which part of their motor cortex is roughly in charge of different functions. Once we've got one of those maps, we put it up on a screen, we compare that with standard maps of anatomical distribution of different functions to make sure everything seems within the right bounds. We look at maps of blood vessels, because we can't put our arrays in blood vessels either. Then we make a rough estimate of where we think the arrays should go. A lot of this is somewhat participant dependent. So the research goals, and this is true of the BrainGate trial in general, is very research participant dependent. And so we try to meet our participants where they are and figure out what are the important research goals that they're going to be able to help us achieve for people who have conditions like they have. When all said and done, we can, to facilitate our surgeons, we can actually 3D print the brain. So this is a hypothetical 3D printed brain right here. And it has some little sticky notes over some places where you might want to place arrays. So this is over the precentral gyrus, and different colors here are signifying the different anatomical parcellations of the precentral gyrus, where we know things that are more dorsal tend to control the hand and arm. I'm using the medical term, things that are closer to the midline tend to control the hand and arm. Things that are a little closer to the ear on the brain tend to control movement of the face and mouth. And so you could put arrays in, say, these hypothetical locations and possibly decode as someone attempted to either move their hand or speak, do other things like that. So there's a lot of functional imaging, a lot of planning that goes into it. And ultimately, on the day that the arrays are actually placed, then we get guided by the ground truth, which is what the surgeon sees when they're actually trying to place the arrays. And when all's said and done, usually works pretty well. We can also 3D print a model skull to help the surgeon figure out exactly where they're going to perform the craniotomy to place the arrays.
Yeah. You mentioned location before, and you also mentioned the blood vessels. I have to admit, every time I see this, I wonder whether one day we're actually look at somebody's brain in the OR and there's actually going to be sticky notes already there. Right? That hasn't happened yet, but it could happen. The location part, we just, as you said, have a rough idea of where we're going to place these arrays, but then sometimes there's a little blood vessel there. And we make an effort not to put an array through a blood vessel as best that we can avoid it. It's okay if we do, but we try to avoid it. But that means, as you know, that we can, when we get to the OR, we will make small adjustments as to where these arrays, where these recording sensors are being placed. And when we're there, that's okay. We're not, there isn't the type of precision that actually requires us to put an array exactly in the location that we planned before we went into the OR, because there are 80 or 88 or 100 billion neurons in the brain, and there's so much redundancy in the information that can be extracted from these neurons that are nearby each other, that if we need to shift a millimeter north or south or east or west, that is dorsal, lateral, medial, posterior, that's okay. Because whatever information we record is going to be valuable towards allowing, we hope, that person to control a cursor on a computer screen, to control their own arm, or perhaps to even be able to recreate speech as they attempt to speak.
Absolutely. I think it's one of the most interesting things that I've learned since being part of this trial, part of this group. And I think, honestly, one of the most interesting findings that this trial has contributed to our understanding of basic neuroscience is how distributed function really is. If you open up the Introductory to Neuroscience textbook that everyone who takes an undergraduate neuroscience class gets, you see this picture of the homunculus that I described, this notion that function is laid out in a very well-organized way, with the top of the motor cortex controlling the legs, and then moving to the arms, and then finally down to the face. And moreover, that the left hemisphere controls movement of the right side of the body, and the right hemisphere controls movement of the left side of the body. And what we found over and over again is that, in fact, not to be glib about it, but to some extent, everything is everywhere. And with our arrays, they're almost always, I think they're always in the left hemisphere, which is the dominant hemisphere for most people. And yet we're able to understand a lot about the side of the body that we shouldn't understand about, that we can actually ask our participants, all right, we've got arrays in your left hemisphere, so now move your left hand, which in theory should be controlled by the right hemisphere. And we can decode what they're doing with incredible precision. Or for that matter, we have arrays up in the part of the motor cortex that should be responsible for movement of the hand. And if we ask our participants, now try saying different things, we can decode things they're trying to say with arrays that are in their hand area. So there's this incredible distribution of information that you can pick up with these arrays. And it gets back, I think, to a question you asked earlier. Why these arrays, as opposed to, say, for example, EEG electrodes that you put on the scalp. And here it's that same notion that the individual neurons, the individual brain cells, contain just so much information, but it's really quite unique to the individual cells. And as soon as you start zooming out and you lose that spatial resolution, all of the signals become sort of blurred. And the best analogy I can think of is one that I first heard from Sergei Stavisky, another one of our investigators in the BrainGate team. It's like listening to a symphony orchestra. So if you stand outside of the concert hall and listen really carefully, you may be able to tell what piece a symphony orchestra is performing as they play. This is, and sitting outside the concert hall, that's like listening with EEG electrodes on the scalp. These Utah-style arrays, these microelectrode arrays, are like having a high-performance microphone sitting in front of the bell of each trumpet and each clarinet, and in front of the bow of each violin. So you can tell, when the second chair violin played the wrong note, that sort of thing. And to try and decipher what each string on a violin is doing from outside the console would be impossible. But once you're there, you can know exactly what's going on.
Second chair violin never plays the wrong note.
Of course not. No, I'm sorry. I'm thinking about my niece's upcoming concert.
Well, all right. She meant that note. Yeah, no, I think it's a great analogy, and in thinking again about the opportunities to record from different parts of the brain, I think, and I think we agree that as this BCI field evolves, there is going to be value to recording from different locations and different distances from the brain. But in our particular trial, we continue to be focused on extracting as much information as we can to ensure the most intuitive, hopefully reliable, consistent, easy to use, always available data that would restore communication or mobility.
Definitely. And I want to talk about the impact that these technologies have on patients. But briefly before that, Dan, you mentioned how there is research in decoding speech neural activity as well for BCIs. Could you tell us a little bit about that research and how it actually differs from decoding motor activity?
Sure. On the one hand, it sort of doesn't, but I'm happy to sort of to describe how it does work. So why are we decoding speech? What first to motivate the how, and the reason is for communication. So really, one of the primary goals of the BrainGate trial from its instantiation was to provide intuitive, easy to use, always available ways to communicate for people who have difficulty communicating because of neurologic injury or disease. So, for example, someone with ALS who's losing dexterous control of their hands and losing the ability to speak may have a really hard time communicating, and over time may rely on other assistive technologies, things that, for example, monitor their eye gaze and allow them to select letters at a time using their eyes. And these work, but they're slow and they're tiring. And we want people to be able to communicate quickly and easily. And so when this trial first began, the way to do that was to decode the intended movement of the hand, wrist, and arm and give someone the ability to control a computer cursor, to select characters on a screen, to type quicker, type faster. And over time, our understanding of how the brain encodes movement of the hand, wrist, and arm got better and better and better. And we're now at the point where we can ask our participants, imagine holding a hypothetical pen or pencil, and we'll just decode your handwriting in real time. And that's incredible. And with that, we can decode, characters per minute, how many individual letters someone communicate at a rate that's approaching the speed that someone without paralysis can type on a smartphone keyboard. So really great, but it's still not fluent communication. It's not what Leigh and I are, talking in real time. And so to do that, we said, well, we've gotten really good at decoding complex movements. And one really complex movement that is really unique to humans, in some extent, is speech. And so we said, let's see if we can decode the movements that someone's muscles of articulation, the muscles of their face and mouth, are making in real time to figure out what sounds they're trying to produce, and in doing so, the words they're trying to say. And that's really how the speech decoding works. It's an analogy to the sophisticated decoding of other movements that we've done. It's just of a much more complex and frankly less well understood group of muscles because there's really much less, if you will, preclinical data to guide us. So now when we decode speech, we do exactly that. We for participants in whom we're going to be doing speech decoding research, we place the arrays over the part of motor cortex that we think is most important for controlling those muscles of articulation. And then we ask our participants to attempt to speak, and we record the neural activity, and we try to match the neural activity that we're measuring to the different phonemes, which are the smallest individual units of speech. You can sort of think of it, although the linguists and the speech pathologists will correct me, but roughly phonemes are combined to make syllables, which are combined to make words. So if you know at any given point in time what is the most likely phoneme, the most likely sound that someone's trying to produce, you keep track of that, and then as time progresses, you get the next sound, the next sound, the next sound, and you combine those together with some sophisticated AI tools that try to match those sounds to the most likely words that they're trying to create. And with that, you can decode what someone's trying to say. And with the with the right set of machine learning tools, that is the right set of algorithms that find that relationship, it works remarkably well.
Yeah, and you mentioned there at the end the machine learning, which is such an interesting kind of evolution of this research over the past 20-ish years or so. The Josephine, you asked earlier about the neuroscience and what we can learn from individual neurons, and I'm a so to speak card-carrying neuroscientist, and so are you.
Yeah.
Part of kind of a bias, not quite the right word, but a bias that went into this research at the beginning is that we were going to extract what we knew or what we hypothesized should be encoded in the firing rate of individual neurons, or maybe even the power in a certain frequency band of the activity of a bunch of neurons. And that makes perfect neuroscientific sense. And then slowly but surely, computers got a little bit better, computer science got a little bit better, machine learning, artificial intelligence, deep learning approaches began to enter this field. And particularly in the speech decoding realm, and together with our colleagues on the BrainGate team at Stanford and UC Davis and Emory and us, where we are at the moment at Mass General of Providence and Brown, really, and others in the field looking at this type of speech decoding research in a very similar way, the application of what I would call kind of agnostic approaches or hypothesis agnostic approaches to decoding has just really taken hold. So we don't need to have a plan or an idea for how the neural activity is going to be related to an intended phoneme or an intended word that's being attempted to be spoken. We can apply, and you know this a lot better than I do, we can apply these incredible tools to build that map for us between the brain activity and the intended output. And that, you know, for me has been 99% exciting and incredible and somewhat magical. And, you know, I'll get over the 1% that's a little disappointing that the fundamental neuroscience hasn't necessarily been absolutely required for us to kind of make these leaps in neural decoding. But nevertheless, I think both are still going to be valuable.
Yeah. And I'm confident that 1% is going to make a big appearance real soon. I think one thing that another really exciting part about this research, aside from creating tools to help people, is that we learn a lot about the brain, and we learn a lot about speech and language. And we are necessarily, as we're doing this research, developing these tools to helping people, we're learning how the brain does complex things. So while the machine learning tools themselves may be agnostic to the actual encoding, the relative temporal patterns of firing when we're recording from different parts of the brain at the same time, the types of activity that cause a lot of cells to fire or not fire lead to new hypotheses. And so as we're getting better and better at decoding individual sounds, these phonemes, we're also learning a lot about how the brain prepares to produce the next set of sounds and how it puts together different sounds into sentences and how the different patterns of activity relate to more complex components of language. So in addition to predicting what sound is the person trying to make, we can understand, you know, is the next noun that they create going to be modified by the previous verb, or is it going to stand in place for the next clause that's coming? And so we're learning a lot about, if you will, computational linguistics in a sense, and it's creating all sorts of new hypotheses about how the brain organizes information in general that still excites me as a card-carrying neuroscientist.
Leigh, can you tell us about the patient populations that stand to benefit from these technologies or currently benefit from these technologies?
Sure. So brain-computer interface research, as it's been happening for the past 20 years, largely has been focused on helping to develop technologies that we all hope are going to restore communication and mobility. So people have been recruited or can be recruited to our ongoing BrainGate trial, for example, who have either cervical spinal cord injury or brainstem stroke or muscular dystrophy or motor neuron diseases such as ALS or Lou Gehrig's disease or related disorders. Those pursuits are incredibly important, continue to be where we focus, but there are other ways that this type of information harnessed from the brain can be used to restore any number of neurologic functions in the future. I think that other populations who will benefit include people with cerebral palsy, other people who have other types of paralyzing disorders. Also, these types of brain-computer interfaces, they are an important limb, if you will, or component of seizure prediction or seizure suppression type technologies. So there's an available responsive neural stimulator device that records chronically from the brain. It detects an upcoming seizure, and upon that detection can then modulate different parts of the brain in order to suppress that seizure, decrease the likelihood that a clinically evident seizure will occur. That's a type of brain-computer interface. Doesn't require somebody to make a voluntary movement, but it records neural activity, it interprets that neural activity, and it does something useful with it, which in some ways is a very simple but I think effective definition of a BCI. One other population, incredibly important, who I think will, and I hope will eventually benefit from BCI research, are people with mental health disorders, particularly mental health disorders that are refractory to adequate treatment with medication. The mental health disorders, in many ways, far more complex than disorders that lead to an inability to move. But if one can record from a part of the brain that, for example, at one moment in time is working the way that that person wants it to, but then, for example, begins to change in a way that may change their mood from one of relative comfort to either one of depression or one of mania. That, just to state the obvious, that transition happens inside the brain. And if we can find the neurophysiologic signature, if we can find the neural activities that anticipate or predict that kind of change from relative health to a condition that that person would prefer not to be in, then perhaps we can detect that signal ahead of time and we can turn that into a modulation of the neural activity to kind of keep that person where they want to be, whether it's related to mood or cognition or perhaps in the future even memory. So I think the potential value of these implantable brain-computer interfaces is large and could help people with many different types of injuries or illnesses.
A goal that you mentioned for BCIs is the restoration of movement. What efforts are being made to help patients control their own bodies through BCIs or brain-to-computer interfaces?
Yeah, there's a few different approaches that that have been tried and continuing to be developed. One is one could gain control over an external robotic device, such as a robotic arm that may be attached to the side rest of a wheelchair. Somebody can think about the use of their own hand and gets intuitive control over that robotic arm. They may, for example, be able to pick up a thermos and take a drink, which is the image that I have in my mind so repeatedly of just an amazing woman in our BrainGate trial who had a brainstem stroke, was able to think about reaching out with her own arm, that she wasn't able to move to reach out to grab a thermos, and she reached out and took a drink of a cinnamon latte of her own volition, and solely of her own volition for the first time in nearly 15 years. So that's one example of one way that a upper limb function, so to speak, could be restored. Other ways would be to regain control of one's own limb with a version of an exoskeleton, particularly a soft exoskeleton or wearable robotics. One of our colleagues, Connor Walsh, the Harvard School of Engineering, has developed some really amazing soft clothing-like robotics that can be worn, and rather than pulleys and cables, they're really balloons informally, bladders more formally, that can be inflated and deflated in order to re-enable movement of one's own limb. And he and colleagues, including Sabrina Paganoni at the Healy ALS Center here at Mass General, have been working together to gain experience in using these types of devices for people with ALS. So that's another way that somebody thinks about moving their own hand to open and close their hand, for example. That might be possible using soft robotics. Two other methods would be to use a BCI, an implantable BCI, in combination with an implantable stimulator or neuromodulator. One, traditionally called FES, or functional electrical stimulation, could allow for the stimulation of muscles. That is, not only are there electrodes recording from the brain, but those are connected through the decoder to electrodes that could stimulate a muscle directly. And as part of our BrainGate research, together with our great colleagues over at Case Western, we have an example of a gentleman with a cervical spinal cord injury who's able to open and close his hand or flex and extend his elbow just by thinking about those movements. And finally, one other approach would be to connect a BCI to a spinal cord stimulator. or to a stimulator that isn't stimulating the muscles directly or the peripheral, but the peripheral nerves. And in that way, one of the real dreams for this research is, as you're alluding to, for people, for example, with upper limb paralysis, is to reconnect brain to limb, to take this intention to move and reconnect it into actual movement. And there's similar research that's ongoing for regaining control over the lower extremity.
Dan, how do you train a participant to control these devices? And are participants trained on one effector able to easily transition to use another effector?
That's a great question. I would flip the question a little bit. We don't train the participants to do anything. We train our decoders based on what the participant's trying to do. So when we want to allow a participant to use the BCI to, control an effector, we ask them just to move your hand. For example, we want our participant to control a computer mouse on a screen. So just try to move your hand up, down, left, right, and we'll train our decoder to understand what those movements are so that the computer cursor does exactly what you want it to do. We may ask them to try different types of imagery. For most of our participants, their paralysis is rather severe, so they may be more comfortable attempting different types of movement. Moving your hand up, down, left, right, as if you're controlling your computer mouse, is really engaging muscles of your shoulder. We may say, try controlling something like an old-fashioned video game joystick. So now you're really moving your wrist. So they find an imagery that's comfortable for them, and then we train our decoder to do what it's supposed to do based on them just trying to naturally move their body. The question of how easy is it to go from one effector to another is interesting. The short answer is it's relatively straightforward as long as our participant is doing the same thing and we've got all of our code correct. So if we tell our participant, hold that computer joystick up, down, left, right, and now instead of a computer cursor on a screen, we're going to have it control the movement of, say, one of the dimensions of movement of one of these external robotic devices. And it should be the same for them, and we just plug our code into a different device. What's interesting, and one of the things we found, is that it does actually get a little more complicated than that because of the important role of feedback. So seeing the intention of one's actions realized in different ways can change the subjective sensation of how much control you have. And a lot of movement control, as we're learning over the years, is not just the movement itself, but also how it feels to move, how what kind of feedback you get, either sensory or visual, as you perform an action. And so we do adjust a little bit for those sort of things and take those into consideration when we switch from one effector to another. But the short answer is we don't train our participants at all. We just ask them to do.
And I think that one of the reasons that happens, and that it happens so easily intuitively, is if we think about somebody who's able-bodied, who is reaching out with their hand to pick up a coffee cup, and then they put the coffee cup down, and they reach out to pick up something else that's on their desk. At the moment, we have a model of a brain, so I guess I'm going to refer to that. They're different devices that are being manipulated. There are different devices that are being picked up. They're being held in different ways. There's a different balance. There's different weight to it. But the brain is really good at controlling that external device, if you will, which in this case is the arm and hand. And the fact that it's going to be picking up a coffee cup or picking up a model of, in this case, half a brain with some sticky notes on it, or is typing on a laptop, or is throwing a baseball. While these are markedly different activities, the brain is really good at just intuitively doing these things, and not only intuitively doing them, but doing them while the rest of the brain is thinking about something else or doing something else. And that's part of why these implantable BCIs, I think, will be so powerful, is that we're harnessing this truly native ability that exists even after neurologic injury or illness to just control an external device, or to attempt to control an external device. We do our job in decoding that properly, that is training the decoders, training the math properly, then we should be able to restore as a field communication and mobility quite readily.
Leigh, you've mentioned in the past that neural data has much more information than currently meets the eye. Could you talk about how patient current and future patient privacy will be maintained and is kept safe.
Yeah, it's such an important uh, concept and approach for all of us in this neural interface field to be thinking about. So the first we're recording with the purpose of further developing these neuroprosthetic devices. Recording so that we can allow somebody who may be unable to move their hand to move their hand again, or to control an external device just by thinking about the movement of their own hand. But undoubtedly, these neurons, we know that these neurons have other roles and that there's other bits of information that are encoded in there. We won't know that until we look for it, and we may not even know that until computer science and machine learning and fundamental neuroscience kind of continues to advance at the same time. So it's possible that kind of arbitrarily, if we look at the same set of neural data 5 years from now, we're going to learn something that's in there that we didn't even know to look for currently. So an easy example of that, which referred a little bit to the question that you and Dan were just talking about, is if somebody is using our own kind of neural interface system to, this from a Society for Neuroscience abstract from years ago, if they're controlling a cursor on a computer screen, and then we kind of move the screen out of the way and they're controlling a robot arm, but they're otherwise just doing it the same way. This work that Vamsi Chabakula and colleagues and Kevin Huang and others did. We can detect from the brain activity whether they're attempting to control the cursor or the robot arm. Now, that wasn't necessarily something that we were going to know, because they were able to control both. They were able to control both quite readily. But the neural activity itself knew, just to briefly anthropomorphize, yeah, I'll figure out which is the right word in a moment, the brain activity itself, that neural activity had encoded in it some signal that allowed us, after the fact, to determine whether it was the robot arm or the cursor being being controlled. Another way to think about it is that the neural activity may be able to determine whether somebody's reaching out to pick up a fork or a spoon, and maybe even more abstract information, which is really to the point that you're getting to. And your question was about privacy or neural privacy. So I think it's quite reasonable that, particularly if somebody is taking a medication that works on the brain or works on the nervous system, it's reasonable to think that if we have neural recordings, we might, for example, be able to detect whether somebody's taking a particular medication based on the neural activity. To some extent, it should be the case that we can do that, because if that medication is working on brain activity and we're recording neural activity, we should maybe be able to detect that. Whether we really can or not with this particular type of recording approach, I think, is unknown, but at least it's worth posing as a possibility. Making this maybe a little more simple, if somebody right now is using our investigational BrainGate system to communicate with their friend or their family member, it's possible that they would prefer that information to be theirs and theirs alone, between the user of the system and the person that they're interacting with. If they're about to make a social media post with that same information, then it was intended to be public. And on the other hand, if they're speaking, having a confidential conversation with somebody in their family, or maybe with their healthcare provider, that presumably is information that we should go through considerable lengths to keep private, or to only share when that person who's generating those data, if you will, or communicating with their friend, colleague, or loved one, intended to share that information. So yes, you're asking about the types of information that are encoded in neural data and privacy. I think at a first pass, much as we do in medicine every day, the first rule is that information is private and is protected and is confidential until permission is given to share that information more broadly. And I think if we go into either our research endeavors, which certainly is this is the approach that we use in our BrainGate research, and I know that others use as well. If we start there and we do our very best to work with our participants to tell them what we know we can decode at the moment, what we may be able to decode in the future, and then we give them, most importantly, the permission to share those data or not. That's really, to me, a very comfortable way to think about neural privacy. And at the heart of that is telling our participants everything that we know, and then really giving them the opportunity to provide informed consent, to provide these data, to share these data either with other research collaborators or with the public. And I should mention that many of our participants, when asked this type of question, they join the trial not because they're hoping to get any personal benefit from being in the trial, but because they want to help to develop and test a technology that will help other people with paralysis. And sometimes when we ask these questions of our participants, and we're beginning a long, as this answer is, kind of a long explanation of how these data may be used and what may be in them and what may be available, they will often say, just get the data out there. We want to help science. We want to move this field forward. And so it's our job to be conservative in many ways, to make sure that we are only sharing information that we have permission to share. On the other hand, there are going to be a lot of people who will want those information shared quite broadly in order to advance science and advance medicine.
Dan, what were the main findings of your safety and feasibility study of the BrainGate system?
Sure. So about, I guess, 2 years ago now, we decided, well, you've been running this trial for about 17 years. And a lot of people were interested. As a consortium, as a clinical trial, we had the most experience with this technology of any group in the world. We had the most participants working with us for the longest period of time. And there was a lot of, there's been a lot of excitement about implantable BCIs. And we said, well, now seems as good a time as any to share what we've learned about the safety of these technologies, so that other people who are thinking about developing them can sort of a roadmap, an understanding of what they may find. If you will, the bottom line finding is that when done the way that we've done it, which we think is with attention to detail and taking care to monitor for all the contingencies that you have to think of when you're working with implanted technology, is that it's really about as safe as any other FDA-approved implanted neurotechnology, which is a loaded phrase. But there have been other implanted neurotechnologies that have been on the market for some period of time. Things like deep brain stimulators for Parkinson's disease and other movement disorders. The responsive neurostimulators that Leigh referenced that are used to treat people with refractory epilepsy. And basically, what we found is that these technologies are really very well tolerated and very safe. I mean, we place the device obviously in an operating room with sterile precautions, and we take very strict care to make sure that around the incision stays clean and we don't have any infections. But they don't seem to. I mean, and they're designed in such a way that they shouldn't. But they don't seem to have any long-term effects on the nervous system. All of the materials are biocompatible. There's no reaction with the brain. People don't get infected. They don't get scars. The brain keeps working, and we know it keeps working because we can continue to decode movement for years. We've had participants in the trial, a couple of participants over 5 years, who continue to work with us, and their signals work well. And so we say, all right, the cortex in which it's implanted must still be healthy, because here we are recording really high quality signals. So it's encouraging, I would say.
Yeah, and I think I'll just add to that, doing my I think doing my job as to temper
Sure.
what everything I agree with. It's an ongoing trial. It's an interim safety report, incredibly detailed, and Dan just deserves amazing credit. 17 years of data, 14 participants, every event in the trial over that period of time kind of re-adjudicated, looked at again, and reported in as detailed a manner as we and our colleagues could potentially come up with. Incredibly boring paper, just to be clear, which in some ways is a good thing. But this is, as we interact with anybody considering these technologies or considering joining in a trial, this is still surgery. There's still risks that are associated with it. We can't make all of those risks go to zero. One never really can in any device that's being placed surgically. There are device-related adverse events that can happen, and we talk in great detail with anybody who's thinking about joining this trial, really to explain what each of those risks are, at least theoretically, the extent to which they've happened, if at all, in the trial thus far. And it's really that type of kind of risk-benefit discussion or list of risks, and from a study standpoint, understanding that benefit is really for people in the future who may benefit from these types of technologies. Those conversations are not only incredibly important, but they're similar to the conversations that a patient might have with their physician in thinking about an approved technology, that is a clinically approved technology. What are the risks? In that case, what are the benefits? And weighing those two, while it's important for us as clinician scientists to have a real handle on the risks themselves and making the risk-benefit equation one that generally could make sense, it's the individual who's considering each of these technologies that kind of evaluates that from their own perspective, and their measure of potential risk, potential benefit or not, is really the right one for that person.
Okay. Final question for both of you. What are the clinically viable endpoints for your research?
That's a great question. I think the most, I mean, this is a loaded question. You know, usability. I think if someone can use this technology and it does what it's supposed to, they can use it to communicate. They can use their BrainGate device and they can talk with their family. They can do their job. They can write the reports that they need to write for work. They can talk with their healthcare providers. They can be part of the conversation. If a participant in the trial says this is working really well, and we didn't go over sort of the nuts and bolts and logistics of the trial, but most of the research occurs, all the research occurs in our participant's home. Most of it occurs with our research technicians. They're present, turning on the system and running research sessions. And when our participants say, I want to use this without you here, it works better than other things that I've used in the past. This is how I want to communicate. To me, that's a success. That's, I think, the greatest endpoint and a goal that we should be working for with all of our participants.
Yeah, I agree. I think that all of these early feasibility or pilot clinical trials, the first outcome measure, in some cases the only formal outcome measure, is one of safety. And the question is this device safe to be implanted, to be in place for a long period of time, for 6 months, for a year or more? And hopefully that in our ongoing trial, it has been encouraging. Exactly the word that I would use is from the standpoint of this implanted array in our hands in the BrainGate trial and in others as well. Whether it's for our investigational technology or others, after having determined that a device appears to be safe, and at least safe enough to move to a pivotal trial, then the question becomes one of both safety and efficacy. Is in a greater number of people, can we determine that what that person wants to use the device for, can that outcome actually be achieved? Is somebody able to communicate more readily? Is somebody able to control their environment more readily? Is somebody able to interact with their friends, their caregivers, their loved ones in the way that they want to interact? All of these questions, and much more refined than in the way that I just described them, those are the way that we get to clinical trial endpoints for any given trial. But if we take a step back from all that, just as Dan was saying, if somebody's using a technology and they're telling us that it's working better than all the other technologies that are available to them, and that they want to be able to use to communicate, or they want to be able to use it to control their environment, that is a sign that we're on the right path, and that this particular approach is one that will have value. And I'm really excited not only about our ongoing BrainGate research, but about all the other implantable brain-computer interfaces that are being developed, all towards restoring communication, restoring mobility, some of them decreasing the frequency of seizures, some of them on a path to restoring mental health. I think all of these approaches are just tremendously exciting, and ultimately that progress is only made because truly amazing individuals find out about these studies, reach out through either friends or family members to us and or themselves, and say, hey, I want to learn more about the study. Ultimately, I want to enroll in this study, and then give us a chance to work with them in their home, sometimes for years on end, to learn how the technology is currently working and how to make it ever better.
Well, thank you both for joining me today for this amazing conversation. I look forward to learning more about brain-to-computer interfaces in the future.
Great. Thanks so much. Great to see you.
Thanks for having us.